today we will be talking about the citric acid cycle, also known as the TCA cycle. But before we get into that cycle, we have to talk about pyruvate dehydrogenase. Okay, so in the mitochondria, we're going to have two membranes. Okay, so we have an inner membrane and we also have an outer membrane. Now, the outer membrane is very permeable. Okay, so the pyruvate is able to cross the outer membrane, but then when it enters the cytosol of the mitochondria, right, it's going to need something to enter the inner membrane, okay, because again, the inner membrane is not permeable, right? So let me repeat, the outer membrane is permeable to molecules, but the inner membrane is more tough and more rigid, so it's not as permeable. So how does pyruvate enter the mitochondria? Right? Well, pyruvate actually enters the mitochondria using a carrier protein, okay? And essentially, it's called a translocase, okay? So, again, scientists are really creative with their names, right? So, translocase is essentially a taxi driver for pyruvate, okay? So pyruvate enters the mitochondria, but it cannot enter the inner membrane, so it uses translocase, which is a carrier protein, and it inserts itself into the mitochondria. Now, the way pyruvate enters is through a symport fashion, okay? So this is a symport right here, symport entry. What does that mean? Symport means to be in the same direction as another molecule. So notice that when pyruvate enters the mitochondria, it is in the same direction as the hydrogen. So whenever pyruvate enters the mitochondria, hydrogen also enters, and they enter in the same direction. Now, in other classes that you might take for biology, you may be taught anti-symport fashion. So anti-symport is when a molecule enters a cell and one thing exits the cell. So they're kind of going in opposite directions. Here, this is a symport fashion, so don't worry about the other one, right? And you should also know that pyruvate only enters the mitochondria under aerobic conditions. Okay, so pyruvate enters mitochondria. And let's call mitochondria mito under aerobic conditions. Okay, so whenever there's oxygen, the pyruvate, which was created from glycolysis, enters the mitochondria via a translocase carrier protein and begins to transform. Now, what does it transform into? Well, before it can enter the citric acid cycle, the pyruvate must be transformed into acetyl-coenzyme A, right? Because acetyl-coenzyme A is kind of like a universal building block. Whenever you eat a hamburger, that burger has bread, meat, lettuce, tomatoes, pickles, whatever. But the hamburger is made of proteins, lipids, and carbohydrates. So how does your body break all those molecules down to produce energy, to produce ATP? Well, your body breaks down the hamburger into its building blocks. It breaks it down to proteins, and carbohydrates. It breaks it down to lipids. But then your body keeps breaking molecules down. It's a very catabolic reaction. It converts proteins into amino acids, lipids into triglycerides, and carbohydrates into glucose. After that, it takes those little building blocks and it converts them to one molecule. And it converts it to acetyl-coenzyme A, right? Because acetyl-coenzyme A can enter the citric acid cycle and produce energy. So that's what your body does. It wants to convert anything that it can to acetyl-coenzyme A and then put that in the citric acid cycle to produce energy, okay? So this process of pyruvate entering the mitochondria is just to produce acetyl-coenzyme A. Now, how is that produced? Well, this may seem like a simple reaction, but it's not. It actually has 5 steps, and they go into great detail. I'm not going to get into that. I just want you to kind of see the big picture over here. So what happens here is that pyruvate, when it enters the matrix of the mitochondria, so essentially this yellow fluid right here, right? So this is the mitochondrial matrix right here. When it enters that, it's going to be exposed to an enzyme, and that enzyme is called pyruvate dehydrogenase. Okay, so whenever pyruvate enters the matrix of the mitochondria, it is exposed to pyruvate dehydrogenase. It will then be converted, right? So again, we have some reductions going on here, right? So we are reducing coenzyme A with a thiol group. Thiol group is just a SH. So right here, this is a thiol group. So we're going to reduce that. Over here, we're actually reducing this as well, this NAD into NADH, right? So of course, this reaction is facilitated via coenzymes, right? So if you don't have certain coenzymes, you're not going to be able to produce acetyl coenzyme A. Now those coenzymes, the main ones are going to be NAD plus, right? And also HS-CoA. Now you can see that this is going to produce some carbon dioxide right here, some carbon dioxide, and this will produce NADH. And also a free hydrogen. Now NADH, and you're going to see its brother FADH2 later on in this video, they're super important because whenever we're doing the phosphorylation oxidation reaction in the next video, they are going to be used to generate lots of ATP. Okay, so this whole video is going to be focusing on the generation, the creation of NADH, FADH2, etc. Okay, so let's get into the citric acid cycle. I just wanted to show you the before reaction. Okay, I just wanted to show you the generation of acetyl coenzyme A that enters the citric acid cycle. So now we will begin talking about the TCA cycle. Okay, so everything that we've done so far in the previous videos have led to the TCA cycle, right? So glycolysis produced pyruvate. Pyruvate enters the mitochondria. It is transported into the matrix via translocase protein, and then it's converted into acetyl coenzyme A via pyruvate dehydrogenase. And now we will be talking about what happens to acetyl coenzyme A inside the cyclic acid cycle, or the citric acid cycle, sorry. Okay, so essentially, this whole process, this whole cycle is actually amphibolic. Okay, so TCA cycle, right, the citric acid cycle, or the carboxylic acid cycle, sorry, is actually amphiphobic, or amphibolic. It's right here. So we have amphibolic, right? And what that means is that it has properties that is both catabolic and anabolic, right? So we could say is both catabolic and anabolic. So let's kind of focus on the big picture first, and then we can go into each process of how it's made, right? Okay, so the big picture is that you're essentially using acetyl coenzyme A as fuel, right? And whenever you're burning fuel, what do you produce? You produce smoke, you produce fumes, right? And those fumes and smoke in reactions, they're actually CO2, right? So if you light some paper on fire, the paper is the fuel, and the smoke that it produces is CO2. So you can use that example to kind of remember that acetyl coenzyme A is used up in this cycle to produce CO2. We can also say that this citric acid cycle also produces ATP, and it produces some interesting molecules. Okay, so it produces ATP, right? Also some GTP, but they can be interchanged. So yes, it does produce GTP, but it also produces ATP a little bit, right? And produces NADH and FADH too. Okay, so this is actually really important. Okay, so the whole process, the whole purpose of the citric acid cycle is to produce a little bit of energy, but the main reason is to produce these NADHs and FADHs too, right? So what do these guys do? These guys, they have a lot of energy, and they go into the cristae or cristae of the mitochondria, and they react with some complexes. And whenever they react with complexes, they generate a lot of ATP. Okay, so they will be oxidized later on inside the mitochondria. Okay, so this whole cycle is a generation of these two guys right here. Okay, so we need these guys to produce a lot of ATP. Okay, and as a side note, the body also uses the TCA cycle to replenish molecules for other reactions. Like, let's say reaction W needs some isocitrate. So the citric acid cycle produces isocitrate, but then some of the isocitrates can go to other pathways, and they can continue those metabolic pathways in your body. Okay, so the citric acid cycle can be used as kind of like a little factory. So any process that needs malate or oxaloacetate, they can get it from the citric acid cycle. Okay, so it produces these oxidizing agents or reducing agents, right? And it also produces molecules that can be used in different reactions. Okay, so these guys right here are going to be called reducing agents, right? Sometimes I mess up the words, right? But reducing agents because they will be oxidized later on. And finally, because ATP can be generated from the molecules converting into each other, you know, certain molecules can convert into different ones and produce ATP as a result, that is called substrate level phosphorylation. Okay, so ATP can be produced via TCA, and that's called substrate. Well, hold on. Let's see, substrate level phosphorylation. And of course, substrate level phosphorylation also occurred in glycolysis, right? So when some molecules converted into different molecules, they produced ATP. And so that process was called a substrate level phosphorylation. Okay, so it's when ATP is generated by the conversion of one molecule into the other. Now, some differences between glycolysis and the TCA is that glycolysis occurs in the anaerobic conditions and it's linear, right? But the cyclic acid cycle actually is cyclic, obviously, and it actually undergoes aerobic condition. Okay, the glycolysis pathway occurs in the cytosol of the cell, but the TCA cycle occurs in the matrix of the mitochondria. Let's get into the formation of molecules, right? So this is like the huge chunk in this video. Okay, so first of all, we're going to combine oxaloacetate with acetyl coenzyme A, and we're going to be forming citrate. Now, citrate is kind of like the physiological form of citrus acid, right? So whenever you eat an orange, it will be converted into citrate. So this is kind of like the physiological form of citric acid, right? So this can be used in different metabolic pathways. Here, we're going to be creating it. Now, how do we create it? Well, we're going to create it by condensing oxaloacetate with acetyl coenzyme A. Now, how do we do that? We do that by using the citrate synthase. What is a synthase? A synthase actually creates or adds to a double bond, right? So we're either so we're either going to create a double bond with synthase, or we're going to be adding something to the double bond. Okay, so either creates or adds to double bond. Okay, so are we creating a double bond? Let's see, we have a double bond here and here. No double bonds over here. Okay, so we're not creating, but that must mean that we're going to be adding something. Well, what are we adding? We're going to be adding, essentially, we're going to be adding this acetyl group right here into the keto double bond. Okay, so this right here is kind of like a keto group. And we're going to be adding the acetyl to this carbon, right? So as you can see here, this carbon had a double bond to the oxygen, and it also had a COOH, not an OH group, but rather it had a COO group, right? So I know I kind of messed those words up, but essentially this carbon had a double bond on this oxygen, right? So here's this carbon right here, right? Here it is. Notice that this carbon lost a double bond, right? But then we added a CH2 and a COO group, right? So we just added this guy here, that's all. So that process of adding the acetyl group to the carbon is called an it's called an aldol condensation. Okay, so this is an aldol condensation. Right. Okay. So what else is important? Well, this reaction is the only reaction in the TCA cycle that forms a carbon-carbon bond. Okay, only portion of TCA where carbon-carbon bond is formed. Okay, so in the other cycles of the TCA cycle, you're not going to be forming any like carbon-carbon bonds, okay? So the process of converting oxaloacetate into citrate requires the formation of this bond right here, right? So that is a carbon-carbon bond. Very important, right? So let's recap. We're going to be using citrate synthase to essentially create citrate from oxaloacetate. Everything rhymes, right? But this is actually an aldol condensation. Because we're adding this acetyl group into the keto carbon. Now, ask yourself, this reaction, is it an oxidative reaction or is it a reducing reaction? So was our carbon oxidated or was it reduced? Well, actually it was reduced, because look, you can easily tell if a process was reduced if the amount of bonds to oxygen decreases. So carbon has 2 bonds to oxygen, but over here this same carbon only has 1 bond to oxygen. So our bonds decrease, meaning that this is actually a reduction. So this is a reducing, or I guess reduction, right? So reduction reaction. So we can say that oxaloacetate is reduced into citrate. Now we need to convert citrate into isocitrate. Now the conversion of citrate into isocitrate goes through a cis-aconitate intermediate. Here's a fun fact. Usually this actually has a iron, so it has an iron sulfur group next to it, right? And that is going to be called the prosthetic group. So it just kind of helps it form and then helps it convert into isocitrate. That's all you really need to know about the intermediate, okay? So what happens here is that we're going to do a dehydration reaction on the citrate, because look, we have a hydrogen and an alcohol, right? So this alcohol can leave, and when it leaves, it's going to be creating a tertiary carbonyl, which is very favorable. Now this hydrogen can leave as well, and it will form a double bond over here, right? So that's what you see on this intermediate. Now, either the same water or a different water molecule can react with the intermediate, and actually form kind of like the opposite, right? So what I mean by the opposite is that this right here is a tertiary alcohol, right? So this alcohol is next to 1, 2, 3 carbons, right? So this alcohol is attached to a carbon, and that carbon is next to 3 carbons right here. So this is a tertiary alcohol. Very favorable. That's why it wants to react, right? If you remember from organic chemistry, tertiary alcohols can easily leave the molecule to create a double bond, right? But over here, when it comes back, this actually forms a secondary alcohol because this carbon that it's attached to is next to 1, 2 carbons, right? So this is actually secondary alcohol. Okay, so essentially all you need to remember is that citrate is a tertiary alcohol, which is very favorable. That's why we're creating citrate, and we're going to be creating isocitrate, right? So isocitrate again is a secondary alcohol. Now, what else can be said about this reaction? Well, this reaction, what kind of is it? Was it a transferase reaction? Was it changing stereochemistry? What was it doing? Well, it was changing the molecular conformation, right? So what changed? What changed was the placement of hydrogens and oxygens, right? We still have the same amount of carbons, we still have the same amount of oxygens and hydrogens, they're just placed differently. So we can say that this is called an isomerization reaction. Okay, so this is isomerization reaction. Right? Did we use water to cut any bonds? Did we do that? No. So we can say that this is a non-hydrolytic reaction. Okay. So essentially we isomerized citrate into isocitrate so that it could be further oxidized, right? So we didn't lose any well, how should I put it? Well, that's for later, but essentially we can further oxidize this molecule into its brother, alpha ketoglutarate. So let's talk about that. So this reaction is very important, okay? So please pay attention because a lot happens in this reaction, a lot of information right here. So isocitrate is going to be converted into alpha ketoglutarate, okay? But I want you to focus on the second carbon right here. So this is the first carbon, this is going to be the second carbon right here. Now notice how many bonds does this carbon have? This carbon has, well, specifically how many bonds does carbon have to oxygen? Well, carbon has 1 bond to oxygen right here. But eventually we will have this carbon having 2 bonds to oxygen. So what does that tell us? It tells us that this reaction is oxidative. This carbon was oxidized. Now this is the first, literally the first reaction in the TCA cycle that is oxidative. So we can say that this is the first oxidative reaction in the TCA cycle. What else is important about this? Well, it's a key process in the TCA cycle. Why is that? Well, this is metabolically irreversible, okay? So if the body, or I guess if the system said, hey, we don't really need to produce ATP, we don't really need to produce NADH, right? Well, it can't just take it back. It can't reverse the processes, right? So before you can convert isocitrate into citrate, that's why you have these little baby arrows, right? So one arrow is going this way, the other arrow is going that way. So that's fine, right? But isocitrate to alpha ketoglutarate is actually metabolically irreversible, okay? So you cannot take it back. We can say that this is metabolically well, I don't know how to spell, so I'm just going to say it's irreversible, okay? Because your boy does not know how to spell. So we're going to say irreversible. Okay, no big words. So, what else is really important in this reaction? Well, how should I say? It's one of the first reactions that produces NADH. Okay, so you know that this is an electron carrier, I guess. This molecule is going to go into the mitochondria cristae and create ATP. So it's actually one of the most important reactions because it produces NADH. Okay, so let's write that in red ink. So it produces NADH. Right. So over here we have NAD. That's a coenzyme. It's going to react and it's going to form NADH and also some carbon dioxide. Okay. So what happens here? What happens here is that the hydride ion, right? So the hydrogen atom on the second carbon is going to react and is going to essentially bond to the NADH, right? Or, excuse me, the NAD. So let's recap. Whenever NAD comes in contact with isocitrate, the hydrogen is going to be really reactive, is going to leave the carbon as H negative, meaning a hydride ion, not very common, but it happens, and is going to attack this NAD right here, and that's going to form NADH. Okay, so that happens, and now we form this carbonyl right here. So we have a C double bonded to the oxygen. Okay, now this free oxygen, this free hydrogen is going to react. Okay, this free hydrogen came from this guy right here. Okay, now it's going to attack this carboxylic acid right here, or I guess carboxyl, right? So let's actually write it out. Okay, so this hydrogen is going to attack this guy, and he's going to come off of the molecule. Now whenever that happens, it's going to form alpha ketoglutarate. Okay, so let's take a step back and let's recap, right? So first of all, isocitrate comes in contact with NAD. The hydrogen on the second carbon becomes excited and it leaves the carbon to bind with the NAD. This forms NADH. Okay, so this is one of the first steps that produces NADH. Very important. Remember that. Okay, so when that happens, this hydrogen atom on the oxygen, right, is going to come off, and this carbon is going to form a double bond with the oxygen. So that's where we get this little hydrogen atom right there, right? Now this hydrogen is going to react with the carboxylate right here, is going to decarboxylate oxaloacetate, a lot of rhymes, and essentially we're going to be forming alpha ketoglutarate. So this is also a decarboxylation reaction. Okay, so we can say that oxaloacetate is decarboxylated by C2H, right? So it's decarboxylated by the hydrogen atom on the C2 carbon, right? The second carbon, and this came from the alcohol group right there. So in total, the TCA cycle is going to have 4 redox reactions. Okay, so it's going to have 4 reactions that are oxidative. This is one of those 4 reactions. So this is the one of 4. We can say one of 4 oxidative reactions in the TCA cycle. We will now be converting alpha ketoglutarate into succinyl coenzyme A. Now, this is the second oxidative reaction. And essentially, you're just converting the energy in alpha-ketoglutarate into succinyl coenzyme A, right? So we have this energy that we need to store because right now it's it's not thermodynamically stable, right? So we have all this energy that can be accessed easily, but we need to kind of package it in a nice little package, right? And that package has to be able to react easily but controlled, right? So we want this new molecule to be easily accessible, but we want it to give energy at the right time, right? So what happens is that we want to convert alpha-ketoglutarate into succinyl coenzyme A because of the thioester. Because of the thiol group of coenzyme A right here. So we can say that the thiol group in how should I say, the succinyl coenzyme A is very high energy. Okay, so this can react with controlled pathways and produce a lot of energy and allow us to further continue the cycle. Okay, right now we need to essentially add a plug, right? So this thioester group that we're going to be adding is a plug, and that plug can attach itself to different metabolic pathways and supply energy. Okay, so that's all we're doing. We're just adding a thioester group to this molecule. Now, to do this is going to be using the alpha-glutarate dehydrogenase enzyme. Okay, so it's going to be using a complex. So a complex enzyme is just an enzyme that uses multiple enzymes to react, right? So this has a bunch of enzymes, but we call that family alpha-ketoglutarate dehydrogenase. Okay, so it's a whole crew of enzymes that react with this molecule to produce this product. But right now we're just looking at the big picture and say, hey, it uses a family of enzymes, and we're going to call that alpha-ketoglutarate dehydrogenase. Technically, it's a complex, right? So that's all it is, right? Uses a dehydrogenase complex. So we're just going to call this alpha-ketoglutarate dehydrogenase complex. So again, that's just a family of enzymes, right? So it does that. So yeah, that's essentially all you need to know. We're just adding a thiol group to this molecule, and that allows it to give energy easily. It also produces NADH, right? So it's the second process that produces NADH. So what is our count? So far we've made two. We've made two NADHs. Okay, so we haven't made any ATP, we haven't made any GTP, we haven't made any FADH, two. We've only made two NADHs, and we've also made two carbon dioxides. Excuse me. All right, so you remember that thiol group that we added in the last step, and I said, well, it's acting like a plug that can produce energy. Well, that's what's happening over here. Right here, this thioester group is going to be reacting with the GDP. Now, you probably heard me saying thioester or thiol group. It's essentially the same thing, so don't worry. Maybe wherever you're at a different university is called thioester, at another one is called a thiol group. It doesn't matter. All that matters is that the acetocoenzyme A that we added is going to be reacting with the GDP, okay? And whenever it reacts with GDP, it's going to produce GTP or ATP. because in some bacteria and in some plants, ATP is produced, okay? So we can actually change this. We can say that this is ATP. So this is the first step that produces energy directly. Now, what is that called? Whenever you're producing energy when you're moving from molecule to molecule. That is called substrate level phosphorylation. All right, so this is the first substrate level phosphorylation. First substrate level phosphorylation. And I know that you want me to draw a happy face because it is a very big deal. Okay, so we're going to be drawing a happy face right there. Let's do that. That's very good. Very happy. So again, all we're doing is that we are reacting the thiol group with GDP. And this happens through Succinyl-coenzyme A synthase, right? So we're going to be synthesizing, we're going to be creating a new molecule, okay? So notice that in succinate, which is the product, we don't have the acetocoenzyme, right? We don't have that. Instead, we just have the carbon and the oxygen, right? So we have COO. So that's all that happens. I mean, it does add an oxygen to this carbon because look, we have two oxygens right here, we have two oxygens right there, and you know, that's fine, but that's all that happens. And you should know that this is the only step in the TCA cycle that produces ATP. Okay, so it's the only step in TCA that produces ATP or GTP, right? So look at this picture right here. Do you see any ATP being produced except from this one? No. So this step right here is the only step that produces energy via substrate level phosphorylation. The other ATPs are going to be produced in the phosphorylation oxidation reaction, right? And that's in a later section of this chapter. Now we will convert succinate to fumarate, right? So before I get into it, this process actually occurs in the membrane of the mitochondria. So this occurs, let's write it down, occurs in the mitochondria membrane. So you're probably saying, well, Brian, where does the other process occur? Where do the other processes occur? Well, these guys actually occur in the matrix. And this occurs in the membrane. Of course, everything occurs in the mitochondria. Okay, so don't worry about that. So that's fine. What else can I say? I can say that this is an oxidative reaction. So this is actually the third oxidative reaction. Okay. Now, why is that? You're probably saying, well, you know, I don't see any carbons that are gaining bonds to oxygens. Well, you're right. I commend you for that. Absolutely beautiful observation. However, notice that these two hydrogens are going to leave the molecule. And when they leave, they're going to be forming a double bond. Remember that oxidation occurs whenever a carbon is gaining bonds to oxygen or it's losing bonds of hydrogen, right? So whenever you lose hydrogen, you're becoming oxidated, right? So you are oxidized, right? So the loss of hydrogens is oxidative, right? So this is pretty important, right? So what else can I say? Well, over here, we're going to be creating FADH2. In the picture, it is written as QH2, Some textbooks do that. However, in this class, we're going to be calling it FADH2. Now, this is a redox reaction. So it's a reduction-oxidation reaction, where half of the reaction is reductive and the other half is oxidative. The process of losing hydrogens is oxidative. So what is the reduction part? The reduction part comes from the FAD gaining 2 hydrogens. Okay, so the gaining of hydrogens is what? It is reductive, right? I don't know why I had like a pause there. Weird. Anyways. So this gain of hydrogens is a reduction, right? So we have gain of hydrogens. Call that reduction. This is the loss of hydrogens. We call this oxidation. So again, redox reactions are like coins. Each coin has a face and a backside, right? So each reaction is going to have an oxidation and then a reduction, right? Now remember that your body is very, very lazy, okay? It wants to maintain the smallest amount of energy possible to do reactions. Now notice that the process will always create a trans fumarate. Okay, so technically, if you want to get really specific, you can say that this is trans fumarate. Why? Well, imagine if I had my carboxyl, or I guess my carboxylate group on this side, right? So imagine the COO on the left side. Well, right here, you're going to have steric hindrance, right? So steric hindrance is when the oxygens are too close to each other and they're going to be repulsive, right? Because they have a negative charge and you don't want to have 2 of the same charges together, right? So it's kind of like having magnets. If you have the south pole of a magnet near the other south pole of a magnet, they're going to be repulsive. They're not going to want to come into contact. And that repulsion drives up the energy. So we don't want that. The body wants to create a product that is stable and has the least amount of energy in it. So it's going to have it in a trans conformation because then the repulsion is going to be minimized. Okay, so this is actually a very stereo-specific reaction. Okay, so this is a stereo-specific reaction. Right? So it's always going to be forming the trans fumarate. Of course, some people just call it fumarate. I get it. If you want to be specific, you can say trans fumarate. Totally fine. Now, well, here's a fun fact, right? Malate, right? Or I guess malonate, sorry. Malonate has a very similar structure to succinate, right? Well, I guess you could say is similar to succinate. And so is going to be acting as a competitive inhibitor to the enzyme. But what enzyme are we using? We're actually using succinate dehydrogenase. Complex. So again, a complex is just a family of enzymes that allows us to convert one molecule into the other. So it looks like a very simple step, but there's a whole family of enzymes that help facilitate this reaction. Now, malonate has a very similar structure to succinate, and so it's going to be acting as a competitive inhibitor to the enzyme, right? So it's going to fit into the succinate dehydrogenase complex enzyme, and it's going to inhibit the production of trans fumarate. Okay, so on the exam, if they, want to make it difficult on you, they're going to say, well, why is malonate very important in the TCA cycle? Well, if you were doing an experiment and you didn't want to produce any products in the TCA cycle, you can just inject malonate into the system and it will actually prohibit the production of fumarate, and therefore the rest of the reaction will not go into completion. Okay, so that's a very fun fact, I guess. So again, we're going to be using the Succinate dehydrogenase complex enzyme. I think that this is one of the most simple reactions in this cycle, because all we're doing is that we're going to be adding water to this to this trans fumarate. So I don't really like this picture that they had in the textbook, but essentially this is a trans fumarate right here. So this is a trans fumarate, and we're going to be adding water, and this water is going to be facilitated by fumarase, right? So again, scientists don't really know how to make names. I hope that they never have to name a baby, because really these names are simplistic. Look, ACE just means enzyme, and fumar is just fumarate, right? So this enzyme for the fumarates, right? Very, very simple. So again, we're going to be adding water. Now, when water adds across the double bond, notice that this is OH and this is H. Together they make water. Whenever we add water across the trans double bond, we're going to be adding alcohol to the opposite end, right? So this is a very stereospecific reaction. Right? So it's a very specific reaction right there. What does this form? Well, this malate actually forms what? It forms an L isomer, right? So this forms L malate. Well, that must mean that this carbon right here is actually a chiral carbon. And, it is a chiral carbon, right? So I think that's pretty cool. And so yeah, that is chiral carbon right here. Okay, so L-malate is formed, and of course this is a stereospecific reaction, and essentially it is facilitated by the enzyme fumarase. So now we go into the final step of the TCA cycle. Now this final step is actually the oxidative reaction. This is the fourth, so it's the fourth oxidative reaction. Okay, and of course this right here is going to be a redox reaction. So this NAD is going to get hydrogen, and that is going to be reduced, right? So this is a reduction. So of course there are 4 redox reactions in this pathway. Okay, so very, very cool. So this is the 4th oxidative reaction, and what happens here? Well, what happens here is that we're going to be regenerating oxaloacetate, right? And we do that from L-malate. So we do that from L-malate. So what happens here is that the NAD plus comes into contact with L-malate. Now hydrogen is going to leave the carbon right there, right? It's going to leave the carbon, and it's going to attack this NAD. Now it forms NADH, and when that happens, essentially this hydrogen also leaves, and it forms a double bond right here, right? So oxygen is going to form a double bond with carbon, so you see that occur right there. This free hydrogen is going to be roaming around in the system, as you can see over here, right? So look at the reactant and look at the product. Notice that carbon only has 1 bond to oxygen, but in the product, carbon has 2 bonds to oxygen. That what makes it oxidative, right? So that is what makes the reaction oxidative. Of course, this occurs because we have malate dehydrogenase. Okay, so that's another enzyme that will facilitate this reaction. So the enzyme used is malate dehydrogenase. So let us quickly recap what happened in this monstrosity of a cycle. So how many steps are there? Well, let's count. We can say that this is 1, this is the second step, this is the third step, the fourth step right here, fifth, sixth, seventh, and eighth step. So in total, there are 8 steps in the TCA cycle. Now, which steps right here are actually dehydrogenase complexes? What steps produce the electron carriers? See, always ask yourself when you're studying, how would the professor ask me these questions? They're not going to be using simple speak, right? They're not going to be talking in simple words. They're going to be talking in academic language. So which steps right here actually create the electron carriers? Well, the electron carriers are as follows. The electron carriers are going to be NADH, right? So that's an electron carrier, and also FADH too. Right, so which ones produce the electron carriers? So let's see, step 3 follows the dehydrogenase complex, so that produces NAD, so we can say that step 3 does it. Let's see over here, we also have another dehydrogenase, right, so that produces NADH, so we can say that's step 4. Let's see what else. NADH is also created by a dehydrogenase in step 8. So specifically, the electron carriers of NADH are produced in step 3, 4, and 8 via dehydrogenase reactions. That's an H. Let's do that. Now you're probably saying, hey, don't forget that there's another dehydrogenase reaction. Yeah, I haven't forgotten that. I never forget that. Okay. So specifically, step 6 is the only step that produces FADH2, right? So step 6 produces FADH2. And of course, this is another dehydrogenase reaction. So what can you say? What can you get from this lecture that I've given you? Well, notice that whenever a dehydrogenase reaction occurs, a redox reaction happens, and whenever a redox reaction happens, the creation of electron carriers also happens. So we can say that whenever you say a dehydrogenase reaction occurring, you can say, well, a redox reaction occurred, and we made electron carriers. Okay, so whenever you see dehydrogenase reactions, you're going to say, well, I know that either NADH or FADH2 is going to be made, and I know for sure that this is going to be an oxidative reaction. So notice that there are only 4 steps, 3, 4, 8, and 6. That's 4 steps. Each one of them are going to be oxidative. And if they're oxidative, they're also going to be reductive, right? So yeah, something is oxidized in that reaction, but this right here is going to be reduced. This right here is going to be reduced, right? Because this came from NAD+, and this came from FAD. So whenever you add hydrogens, you're going to be reducing, right? So we can say that this is reduced to, right? So NAD+ is reduced to NADH, FAD is going to be reduced into FADH2. Now, in what steps is carbon dioxide formed? Well, carbon dioxide, they're formed in succession, right? So if you form one carbon dioxide, all you need to know is that the other carbon dioxide is going to be formed afterwards, right? So you just need to remember that carbon dioxide is formed in step 3, right? So CO2 is formed in step 3, and then immediately afterwards carbon dioxide is going to be formed in step 4. Okay, so they form in succession. Another step that is very important is going to be the 5th step, because it generates energy, right? So we can say that it generates energy. So let's do that. We can say that generation of energy only occurs in step 5. Because step 5 is going to produce ATP or GTP. Usually ATP is produced in plants and in bacteria. Okay, so animal cells typically form GTP, right? So the GTP or the ATP is formed through the thioester of the coenzyme A. Okay, so we can say that ATP or GTP is formed via substrate level phosphorylation. So what are the most important steps that you need to remember? Well, the most important steps in the TCA cycle would have to be, in my opinion, steps 3, 4, 5, 6, and 8. Okay? So even if you don't know what happens, like, oh, well, I don't remember that citrate converts to isocitrate. I don't remember that. Well, just know that in step 3, this is going to be metabolically irreversible. Irreversible. Let's see, reversible, right? And this also produces NADH. Well, step 4 also produces NADH as well. This one produces ATP or GTP, depending on which cells you're working with. This guy right here produces FADH2. And this guy produces NADH. So already we've created 3 NADHs and we've created 1 FADH2, and we created 1 ATP. So we can say per pyruvate, how many electron carriers are we gonna produce? Per pyruvate, we produce 3 NADH in the TCA cycle. Per pyruvate, we created 1 FADH2. Per pyruvate, we created 1 ATP or GTP. Now, if you were very observant in the beginning of the video, you would notice that an NAD was reduced into NADH whenever pyruvate was converting itself into acetyl-coenzyme A, right? So in the beginning, I said, hey, we're going to be using pyruvate dehydrogenase. Well, that created an NADH. So technically, it produces 1 NADH in the pyruvate dehydrogenase. Okay, so this actually occurs prior. So this is pyruvate dehydrogenase. Okay, so in total you make 4 NADHs per pyruvate. 3 of them are formed via the cyclic acid cycle. 1 of them is formed in the pyruvate dehydrogenase reaction, and we also form 1 FADH2 and 1 ATP or GTP. Let's talk about regulations and how different things can regulate different parts of the cyclic acid cycle. So overall, the TCA cycle can be regulated by the supply of acetyl-coenzyme A, right? Because everything depends on this bad boy right there, right? Because if you have a lot of acetyl-coenzyme A, then you're going to be producing a lot of electron carriers and a lot of ATP. But whenever you have a little bit of acetyl-coenzyme A, that means that you're not going to be producing a lot of electron carriers, right? So the TCA cycle as a whole is regulated by the supply of acetyl-coenzyme A. And also, it's regulated by the products formed. So certain products in the reactions can actually do feedback inhibition, right? So let's just do an example, okay? Let's say that one pathway, or I guess one reaction, produces product Y. Now, product Y is going to go back and is going to prohibit the production of more product Y. Because product Y cannot be produced anymore, the reaction cannot continue. Even if it does continue, it's going to do it slower, right? So it's also regulated by feedback inhibition. Also, the enzymes that are needed to do the reaction, well, if they're being inhibited or they're being modified covalently, let's say like a poison is put into your system and is destroying the enzymes, well, that's going to regulate the TCA cycle. Because if you have no enzymes to react with, then clearly no product is going to be made and no TCA is going to occur, right? So no cycle is going to occur. So it's also regulated by enzymes. Modifying enzymes. Now you're probably wondering, well, what regulates the supply of acetyl-coenzyme A? Because you said, well, sometimes you have high levels, sometimes you have low levels. Well, what regulates that? And the answer to that would have to be the Pyruvate Dehydrogenase Complex. Okay, so the Pyruvate Dehydrogenase Complex regulates acetyl-coenzyme A levels. And if you regulate acetyl-coenzyme A, you're going to regulate the TCA cycle. Therefore, regulates the TCA, right? So you can think of the Pyruvate Dehydrogenase Complex, we're going to call that PDH. We're going to call this PDH. We can say that PDH is going to be inhibited by a certain amount of things, right? And we can also say that the PDH is kind of like the gateway keeper, right? So it's kind of like the gatekeeper, of the aerobic metabolism. So we can say that PDH is the gatekeeper of aerobic metabolism. Right? Because really, how much TCA is going to make electron carriers depends on PDH. If PDH makes a lot of acetyl-coenzyme A, then a lot of electron carriers are going to be made. If PDH makes a little bit of acetyl-coenzyme A, then a little bit of electron carriers are made, right? So what regulates PDH? Well, we have some inhibitors. And the inhibitors of PDH are going to be NADH, ATP, and acetyl-coenzyme A, right? Now, also you can have fatty acids. Now, why are they inhibitors? Well, they're inhibitors, right? Because you don't want to have a buildup of these molecules, right? So again, we're going to have pyruvate is going to convert itself into acetyl-CoA. And what happens here is that NAD is going to become reduced into NADH, right? NADH. Now, if you have too much NADH, well, that kind of shows that you're producing too many acetyl-coenzyme A's, right? because if you have too many NADHs, well, you're not using your product. As you continue to use up your acetyl-coenzyme A's, your NADHs are used up as well, and therefore your NAD has to be used to create more NADHs, right? So if you have a buildup of this guy right here, then clearly you're not using this guy. And if you're not using acetyl-coenzyme A, then obviously you have a lot of energy, right? You have a lot of ATP. So if I have high amounts of ATP, I'm not going to be using a lot of acetyl-coenzyme A. And if I'm not using acetyl-coenzyme A, then why am I producing a lot of NADHs? I don't need to produce a lot of NADHs if I'm not using acetyl-coenzyme A, right? So these are kind of inhibitors. If you have high amounts of NADHs, you're not going to convert pyruvate into acetyl-coenzyme A because clearly the system doesn't need to create energy. If you have high amounts of ATP, we don't need to create acetyl-coenzyme A. If we have high amounts of acetyl-coenzyme A, then we don't need to create more of it. Now, the reason why fatty acids are also inhibitors is because fatty acids can convert themselves to acetyl-coenzyme A, right? So remember, that's one of the reasons why we want to convert into acetyl-coenzyme A. Proteins, lipids, and carbohydrates can convert into that molecule. So we talked about inhibitors. Then what are some stimulators? So what are some stimulators? What makes the system create acetyl-coenzyme A? So some stimulators would have to be NAD+, AMP, and what else? Coenzyme A with a thiol group, excuse me. So NAD, if we have a lot of NAD, we need to create more NADH. That's going to drive the reaction forward. If we have a lot of AMP, that means that we don't have a lot of ATP. So we're going to do the reaction. If we have coenzyme A with a thiol group instead of an acetyl group, then we're going to convert this guy into acetyl coenzyme A. So these are stimulators and these are inhibitors. So think of the stimulators as kind of like the opposites of the inhibitors, right? So NAD is going to be reduced into NADH, AMP is going to become ATP, and coenzyme A with the thiol group is going to become acetyl coenzyme A. So now let's talk about some inhibitors of some specific parts of the TCA cycle. Let's begin with citrate synthase. Okay, so what is the inhibitor of citrate synthase? So if we go over here, we can say that an inhibitor, well, obviously the inhibitor would have to be citrate, right? Because if you have a lot of citrate, then you're not going to need to make more citrate, so that part of the reaction is not going to occur. Of course, this is a feedback inhibition reaction, or not reaction, but feedback, right? So feedback inhibition, right? So that's citrate. Another inhibitor is going to be called NADH right here, right? So obviously, if you have a lot of NADHs in the system, you're not going to do any of the reactions in the cycle. So for the next couple of pathways, right, when I'm talking about inhibitors or stimulators, NADH is always going to be part of the inhibitor list, right? Because if we have a lot of NADHs, we're not going to need to do the cycle, because the whole purpose of the cycle is to create NADHs and FADHs too, right? That's all we're doing. So there you go. Another inhibitor would have to be ATP, and another one would have to be Succinyl-CoA, right? So these are inhibitors for the citric synthase. And a stimulator would have to be the following. It would have to be ADP, right? So ADP is actually going to be a stimulator. If you have ADP in the system, then you're going to create citrate, because clearly you used up some ATP. That's why we have ADP, and we need to replenish the ATP that was lost. So we need to create more citrate and start the cycle. So that is a stimulator for the citrate synthase. Now, what are some inhibitors for this pathway, for the isocitrate dehydrogenase? the inhibitors always start with NADH, right? So the inhibitors would have to be NADH and also ATP for very simplistic reasons. Again, if we have NADHs and ATPs in the system, then we don't need to create more energy. We don't need to do the cycle. So we don't need to do isocitrate dehydrogenase reactions. Okay, we don't need to do that. But we do have a lot of stimulators for this one. So we have stimulators. We do have stimulators, and that is the NAD plus. We also have ADP and also calcium. So notice that these are opposites. These need to be reduced into NADHs. This one needs to be phosphorylated to create ATP, right? So we need to add a phosphate group to this guy to perform ATP. Okay, so if you have a lot of NADs right there in the system, we need to create more product to form NADHs. If we have a lot of this guy, we used some ATPs in the reactions and we need to replenish the ATPs, so we need to do the reaction to form ATP, right? And of course, you may not know this, calcium is needed to do this reaction. So it's kind of like a facilitator for the enzyme, okay? So that's always important to know. Calcium is needed to do isocitrate dehydrogenase reactions. But now, let's talk about the alpha-ketoglutarate dehydrogenase. What are some inhibitors? So some inhibitors are NADH, ATP, and Succinyl-CoA. So that is a Y, right? So that's NYL-CoA, right? So again, these are the inhibitors because of the reasons that I've already told you, right? I've told you like twice already. So the stimulators, let's put that M closer to the I. They're going to be NAD plus ADP and AMP. So really, this kind of is really dependent on the amount of ADP and AMP. Of course, if you used your ATP to do reactions, you're going to be producing ADP and AMP, and that's going to be driving the reaction to create more ATP, right? So there you go. The stimulators would have to be NAD plus, ADP, and AMP. And you're probably wondering, well, you know, if we keep doing this cycle, aren't we eventually going to be running out of, products? Like, if we keep doing this reaction, aren't we going to run out of alpha-ketoglutarate? Are we going to run out of succinate? Are we going to run out of fumarate, right? So how do we replenish those molecules? Well, remember in the beginning of the video I said, hey, a lot of processes occur in the mitochondria. The TCA is connected to other metabolic processes. Well, that applies here. If we kept doing the cycle, you're right. Eventually our products would run out and we would have to create more products. But how do we create those products? We create those products by actually having amino acids. So we have amino acids that enter the cycle at different parts, right? So that kind of helps out a little bit. Right? And then not only that, but we can also use fatty acids, right? We can also use fatty acids. So whenever we want to make fatty acids into a different molecule, specifically if we want to convert fatty acids into acetocoenzyme A, we would have to do a specific reaction, and that reaction is called beta oxidation. So to recap, in order for this cycle to keep going and not run out of molecules to work on, we have to rely on amino acids, different amino acids like alanine, phenylalanine, and different amino acids entering the cycle at different points to replenish the molecules that are being used up. we can also rely on fatty acids. And fatty acids can be beta oxidized into acetyl coenzyme A, and that can actually help drive the reaction forward. So we can have some fatty acids that will convert into acetyl coenzyme A and help maintain the cycle. Right? So that is the end of this video. I hope that you understand the TCA cycle better and that you do well on your exams. Remember that I always believe in you, and thank you so much for spending your time with me and watching this video. So I hope you have a great day and that you stay safe. All right. Love you.